Hello and welcome to the next module in Waters' Peptide and Protein Bioanalysis Bootcamp. My name is Kelly Doring and I'm a Senior Business Manager at Waters Corporation. Today I'll be presenting on ligand binding assays and LC-MS for peptide and protein quantification. The two primary technologies used for absolute quantification of small and large biomarkers in therapeutics are ligand binding assays and LC-MS. The choice of which technology to use is dependent upon multiple factors, including existing knowledge of the analyte and its metabolites, how far along a candidate is in the pipeline, your team's level of expertise, availability of reagents such as standards and antibodies, and the resources available for reagent and method development. In this module, we will discuss LBAs and compare the advantages and limitations of this technology when compared with LC-MS. We'll then present 3 examples of when LC-MS is the preferred method for quantification of insulin analogs based on specificity requirements and the need to multiplex. Both technologies we're discussing today can be used for the quantification of both small and large molecules and biological matrices. For the purposes of this training series, we're focused on the bioanalysis of peptide and protein-based biomarkers in biotherapeutics. The ligand binding assay, or LBA, is often considered a secondary detection method because it relies upon antibody or antigen affinity for a target analyte, whereby a concentration of that molecule can be determined based on chemical signal intensity relative to the signal intensity of standards in a calibration curve. As we will learn, there are a lot of factors to consider to ensure that the detection reagents are binding to the target specifically. Liquid chromatography mass spectrometry, or LC-MS, is a method that directly measures the analyte based on its mass-to-charge ratio. The relative intensity of the peak area specific to the analyte's M over Z value is compared to a calibration curve to arrive at its concentration. As will be discussed in detail in other modules, LC-MS harnesses the power of selectivity through LC separation coupled with mass detection. The terms LBA and immunoassay are often used interchangeably and encompass the general premise of leveraging antibody or antigen binding to a target analyte in order to measure the concentration in a sample. Typically, these methods are grouped into 2 main categories: the radioimmunoassay, or RIA, and the enzyme-linked immunosorbent assay, or ELISA, also known as the EIA. The ELISA combines antibody binding with enzymatic detection, whereas the RIA uses a radioisotope. The first immunoassay was an RIA, developed in 1959 by Drs. Rosalind Yalow and Solomon Berson to measure insulin in diabetic patients receiving insulin treatment. This discovery earned the team the Nobel Prize in 1977, but even more importantly, the RIA has served as the foundation for many of the other immuno-based assays that have been developed since, including the first ELISA, developed in 1971 by research groups in the Netherlands and in Sweden. The RIA, by design, is a competitive immunoassay, whereby the radio-labeled analyte is combined with a sample and an antibody specific for that analyte. The endogenous analyte competes with the radio-labeled analyte for binding to the antibody. And in the end, the radioactivity counts that are measured are inversely proportional to the amount of analyte in the sample. That is, the greater the amount of radioactive analyte remaining means there was less endogenous analyte present. These assays are typically performed in single tubes that can be counted in a scintillation counter. Sandwich assays rely upon a pair of antibodies to bind to the analyte of interest, and this is what we see for the majority of ELISAs that are developed because it increases the specificity of the assay. In a sandwich assay, the capture antibody is typically bound to a solid support like the well of a plate or on a bead. The sample is added to the well and the analyte of interest is bound. A secondary antibody tagged with a substrate is then added to the sample and the substrate is activated by an enzyme, which yields a chemical signal that is directly proportional to the concentration of the analyte of interest. Typical ELISA formats are 96 or even 384 well plates, and these can be read by a microplate reader. But some of the newer platforms use formatted CDs with microfluidic channels. ELISAs can be built as competitive assays as well. Whether to build a competitive or a sandwich ELISA depends on the reagents available, the size of the molecule, and whether two antibodies can effectively bind unimpeded by steric hindrance. Although RIAs are still in use today, they're used much less frequently than ELISAs because of the desire to minimize the use of radioactivity, the need to use smaller sample volumes, and the push for even greater sensitivity. As we move on in our discussion, the focus will be around the ELISA. As I'd mentioned, the original RIA has proven instrumental in the development of newer immuno-based technologies. Where the RIA requires a radioisotope such as 125 iodine, ELISAs now use a variety of substrates, many of which are listed here. The most common are chromogenic substrates like TMB or tetramethylbenzidine that require a simple UV plate reader. However, other substrates can be used to push to lower limits of quantification and to enhance the dynamic range of the assay beyond a typical 1 to 2 orders. Multi-mode plate readers are now available that are equipped with the detectors for a range of readouts, including fluorescence and chemiluminescence. Other systems are also available that allow for multiplexing and/or single molecule detection, and a quick search on the internet will bring up a variety of these newer offerings. These platforms come with differing levels of built-in automation to afford higher throughput capabilities, and even with the traditional colorimetric ELISAs, labs have options to bring in automation and to minimize the hands-on time required. The use of antibody-analyte binding technology goes beyond what is used in LBAs. For example, it is employed for a host of histopathological and molecular biological techniques, including immunohistochemistry, Western blotting, and others. Pertinent to our discussion here, the technique can also be used as a means of isolating an analyte of interest from a sample, also known as immunoaffinity. This practice helps minimize potential interference that could hamper the subsequent analysis of the sample. Using immunoaffinity for sample prep prior to LCMS is becoming increasingly popular, especially when the analyte sensitivity requirements are high. Hybrid LCMS is the common name for this technique, although it is also referred to as IP LCMS and others. Common formats for performing immunoaffinity specifically include plates, agarose or magnetic beads, columns and pipette tips, all with varying degrees of automation possible. The technique can range from generic cleanup, such as the use of protein A or G, to highly specific antibodies for the analytes of interest. The choice of reagent will depend on the analyte, the matrix, reagents available, sensitivity requirements, and so forth. Even if the capture antibody is not wholly specific, the beauty of LCMS is that LC coupled with MS detection provide additional levels of separation, showing a true complementarity of the LBA and LCMS techniques. There's a long history for the use of immunoaffinity in LBAs, which speaks to the many advantages of the technology. At the top of the list is sensitivity, often in the picogram or nanogram per mil range, small sample volume requirements, their ease of use and throughput capabilities. The technology and expertise level required to run LBAs is relatively low, especially when it comes to the manual and semi-automated options available. And for large molecule quantification, especially for well-characterized molecules, it really is the go-to technology for all of the above reasons. For all of the advantages, LBAs do have some limitations, with the primary watch-out being around specificity. Accuracy of the method relies heavily on the antibodies used and the knowledge of the analyte and how it behaves in biological matrix. If antibodies are developed without enough information about the molecule, there is potential for cross-reactivity with closely related species or circulating fragments, which could impact the accuracy of the results. Although some LBA manufacturers make multiplexing panels available, there's always a concern about cross-reactivity and how fit for purpose these methods will be in regulated environments. LBAs are used in a variety of settings, including clinical diagnostics, and in these cases are fully validated. When it comes to more research-grade kits, however, standardization across kits is often lacking, which can result in lack of reproducibility of findings across labs. In addition, compared with LCMS, the dynamic range of the LBA is limited, which can make PK studies challenging, where there is a wide range of concentrations expected. And if building a new assay in-house, then there's a lot of time and investment that goes into generating both the reagents and the method compared with an LCMS method. However, accuracy and reproducibility of the LBA remains the top concern. This 2015 Nature publication cites cross-reactivity, variability in assay conditions as the most common problems with antibodies used in biomedical research. In one quote, it stated, an evaluation of 246 antibodies used in epigenetic studies found that 1/4 failed tests for specificity, meaning that they often bound to more than one target. Again, this makes the argument for verifying specificity of immuno-based assays even stronger. LBA has been the primary technology for large molecule quantification, but as LCMS technology has evolved, its attractiveness and adoption continues to grow. LCMS allows us to overcome many of the shortcomings of LBA, especially when it comes to multiplexing. And importantly, LCMS can be used to verify the specificity of the LBA. It's unlikely we'll see a complete replacement of LBA by LCMS, but as stated earlier, we're already seeing the use of these technologies in tandem, and that trend will continue as a means to improve selectivity and sensitivity. LBA is especially attractive when there is a demand for higher throughput, such as in late development. Earlier on, less is typically known about the biotherapeutic or biomarker being studied, so developing specific antibodies can be very difficult. In some cases, depending on the molecule, the potential for cross-reactivity to closely related metabolites can be so high that the development of an accurate LBA is impossible. And in these cases, and or when multiplexing capabilities of LCMS are an advantage, LCMS methods can be justified over an LBA, even at later stages of drug development. At that point, it really is all about accuracy. And although we are focused on tandem quadrupole LCMS in this module, please tune into future modules where we will be exploring the role of both tandem quadrupole and high-resolution MS in large molecule quantification. Earlier in the module, I described the first LBA, which was the insulin RIA developed by Yalow and Bearson. Since that time, the incidence of diabetes, especially type 2 diabetes, has reached epidemic proportions. To date, recombinant human insulin and its analogs are perhaps the best known and most widely sold biotherapeutics. Some of the more common insulin analogs used for treatment and depicted here represent both slow and fast-acting therapies that are often given in combination. And what this represents for the pharma industry is enormous market potential, and it speaks to the analytical need for specificity. Historically, insulin has been quantified using LBAs, and these methods, especially ELISAs, are very attractive given their sensitivity, ease of use, and low sample volume requirements. However, these affinity-based assays lack both adequate specificity and the ability to multiplex more than one treatment regimen. So let's move on to our first LCMS example. The work cited here by Erin Chambers et al. In 2014 shows a single method for the simultaneous direct quantification of intact human insulin and multiple insulin analogs in human plasma by LCMS. Intact insulins are challenging to analyze by LCMS for the following reasons. MS sensitivity is low due to poor transfer into the gas phase. Poor fragmentation patterns exist due to the presence of multiple stabilizing disulfide bonds. Distinguishing between human insulin and insulin Lispro, for example, is very challenging because they differ by a simple reversal in the position of two amino acids. Only a single low molecular weight fragment differentiates the two, making selective sample preparation and chromatography critical. In addition, insulin and its analogs suffer from nonspecific binding and poor solubility, making LC in sample preparation method development difficult. This method uses protein precipitation, mixed mode SPE, and a multi-dimensional LC method, including a solid core particle column containing an anion exchange stationary phase. More details on optimizing LCMS method development for peptides will be discussed in future modules, but the highlights of this method are that it could distinguish between insulin in the 5 analog studies, with LLOQs ranging from 50 to 200 picogram per mil, near what one would see for LBAs. As for sample volume, 250 microliters may seem like a lot, but remember this assay yielded results for 6 analytes in one run. The first example was focused on simultaneous quantification of insulin analogs. This next example was published by Merck, West Point, and Inventive Health Canada in 2017. An excellent quote from the article reads, "LCMS techniques provide critically needed selectivity based on the analyte's physicochemical properties, molecular weight, and charge states, and hence offer unique opportunity in addressing the specificity concerns presented with immunoassays." Insulin glargine, also known as Lantus, is a recombinant long-acting human insulin analog, and MK-1293 is the Merck biosimilar. Interchangeability between MK-1293 and the reference product has to be assessed during biosimilar development to ensure no clinically meaningful differences in human pharmacokinetics, or PK. Thus, the target assay required the ability to distinguish between endogenous insulin, glargine, and the glargine's 2 metabolites. Throughout a series of comprehensive evaluations and method optimization, the authors arrived at a hybrid LCMS method as being the best approach to meet their requirements. In this case, compared with example 1, a larger starting sample volume along with immunopurification were used in conjunction with analytical scale LCMS. The highlights of this method show the ability to successfully distinguish insulin glargine and 2 closely related metabolites. This method has been fully validated to meet regulatory requirements and has been successfully applied to support biosimilar clinical studies with thousands of PK samples. The third example I'll discuss also pertains to insulin analogs and is an adaptation of the method shown in example 1. In this method, microflow LCMS was employed to assess how much further the LLOQ could be pushed. Microflow LCMS will be discussed in greater detail in a subsequent bootcamp module, but one of the benefits of microflow in the IonQ system is sensitivity gain due to improved sampling efficiency, ionization efficiency, and decreased matrix effects. The configuration used include a multidimensional microflow LC coupled with an integrated ionization source. The same sample prep was used with lower starting volume than the original method. And the highlights of this method were that in addition to distinguishing amongst insulin and the 5 analogs, an improved LLOQ was achieved, taking it from the original range of 50 to 200 picogram per mil to down as low as 25 picogram per mil across all analogs with 100 microliter starting sample volume. When 50 microliters was used, an LLOQ of 50 picogram per mil was achieved. In this case, sample volumes and resulting LLOQs were even closer to what one would see with an LBA, and it demonstrates yet another LCMS approach that can be used when an LBA is not suitable. I hope that these 3 examples for LCMS quantification of insulin analogs and metabolites help demonstrate the advantages of LCMS quantification when it comes to the interplay between selectivity, sensitivity, and multiplexing. One of the key takeaways from these examples is that there are a variety of sample prep and chromatography approaches that can be optimized along the way. Although the inherent benefits of LBAs, such as ease of use, low sample volume, and high sensitivity make the LBA a very attractive technology for large molecule quantification. They are nothing if the assay does not yield the specificity required to distinguish between closely related analogs or metabolites. So wrapping up this module comparing LBA and LCMS, it can be concluded that choosing the appropriate technology for a program's bioanalytical strategy will depend on the analytes of interest, the reagent availability, and the time and cost constraints. We discussed the benefits of LBA and it being the gold standard for large molecule quantification, but only if specificity requirements are adequately met. Verifying this specificity can be accomplished using an alternative, highly specific technology like LCMS. And this becomes incredibly important as the method is moved into regulated labs. In all, advances in sample prep, chromatography, and mass spec have led to the development of robust, sensitive methods on par with LBAs, and has proven especially useful when looking to simultaneously quantify multiple analytes. I'd like to thank you for joining us today for contrasting LBAs and LCMS for peptide and protein bioanalysis. And stay tuned for more modules in Waters Peptide and Protein Bioanalysis Bootcamp series.